We tend to think of living things as having definite shapes and boundaries. But under the microscope, we find organisms whose very bodies are fluid, shifting and transforming as they move. This video begins our series exploring the world of Amoeboid life, where a single cell can flow, stretch, and reshape itself entirely, a body without a fixed outline. The organisms you are seeing are called amoeboids, not a single branch of life, but a way of living, a strategy that has evolved again and again across the tree of eukaryotes. What unites them all is the same remarkable feature, pseudopodia, extensions of the cell itself that serve as both tools and limbs. Through these shifting projections, the cell can move, sense, feed, and even build. Lobose amoebas form blunt lobes that roll the cell forward. Phyllospores extend slender threads to probe their surroundings. Foraminifera weave networks that merge into delicate feeding webs. And heliozoans use radiating rods supported by microtubules to impale their prey. Beneath these movements lies an intricate flow of cytoplasm, an internal current that shifts the cell's scaffolding of actin and microtubules. This same machinery operates within our own bodies. Human white blood cells use amoeboid motion to travel through tissues and engulf invaders, echoing behaviors that first evolved in ancient single cells. Amoeboid organisms are not rare curiosities, they're everywhere. In soil, freshwater, marine sediments, even on plant roots. They feed on bacteria, algae, and other protists, forming an essential part of the microbial food web. In doing so, they recycle nutrients, regulate microbial populations, and influence processes as large as soil fertility and ocean carbon cycles. Some leave behind shells, intricate tests of calcium carbonate that sink to the seafloor after death. Layer by layer, these shells record the chemistry of ancient oceans, giving us one of the most detailed archives of Earth's climate history. A single cell becomes part of the story of Earth itself. But their importance extends even further back in time. Many biologists believe that early eukaryotes, the ancestors of all complex life, were already capable of amoeboid movement and phagocytosis. That ability to engulf and digest other cells may have paved the way for one of life's most transformative events, endosymbiosis, when a free-living bacterium began living inside another cell, eventually becoming the mitochondria and chloroplasts that power complex life today. Seen this way, amoeboid motion isn't just an isolated adaptation. It represents one of the oldest and most enduring solutions to the problem of movement and survival, a foundation upon which much of complex life was built. In this ongoing series, we'll explore this extraordinary way of life in detail, the many forms of pseudopodia, the ways these organisms feed and build, how single cells can cooperate to form something larger, and what these movements reveal about the shared mechanics of all living cells. This is the world of the amoeboid, simple in form, yet endlessly inventive in motion. Thanks for joining us as we begin this exploration into amoeboid life. Make sure to hit the subscribe button so you don't miss our next episode in this series. And if you have any questions about amoeboids that we can address in upcoming videos, be sure to leave a comment.